A very good morning to my class. This is the chapter 17, protein synthesis. It's also called gene to protein of BIO 101. In the previous chapter, up to that, what we had covered was we talk about DNA being the most important molecule, macromolecule, because it holds all the information. Okay, so we went through a sequence where we tried to understand the structure of DNA, and then the mode with which it replicates, and that how important it is for making anything. Now, in this chapter, what we are doing is we are talking about how this information is actually made into a product. Which means whatever is the gene, that gene is finally made into a protein. Okay. So a sequence of 2-3 slides which will give you a list of important topics that we will cover. Okay, so there was a scientist, his name was Garrett. Excuse me, let me just go back. Yeah. There was a scientist, his name was Garrett, and he is the one who is credited to have made the connection between a deficiency and some defect in the gene, and that is why you have that deficiency, and so you're not able to make the specific product properly. Okay, so and also we would be talking about what is the central dogma or the chain of command, and how from the higher most authority, how it kind of the information filters down, and finally how the work gets done. Okay, so you might have a boss, and then his secretary would have made some memo and then passed it on to a supervisor. The supervisor passes it on to the actual employee or the worker, who will go ahead and make that product. So kind of like that. So this is like a sequence in which the chain of command exists. So you also have a sequence in which a chain of command would exist in your cell, in making a decision or how a final product is being made. Okay, so we also have majorly when we are talking about the process, we discuss how it would be in a prokaryotic cell and how it would be in a eukaryotic cell. Most often the process, the overview is the same. There might be some minor details that would be different in either of these kind of organisms. Then we talk about how a code has come to be, and what is the genetic code, and how it was deciphered. And right now we also use this specific information, and we can kind of engineer genes, we can modify genes, we can add a certain sequence of DNA. The reason being the code is the same, so we can cross boundaries, the species boundaries, meaning gene from a certain creature into a certain plant to give it resistance to something, or gene from the humans that you would be lacking, like diabetics lack in a certain gene wherein they can efficiently make insulin. So in place of that, what happens is you kind of sequence that gene and engineer it so that it gets into a prokaryotic bacteria, and then you grow this in a fermentation tank and extract the product, and there you would get lots of insulin. So you can, the whole process is possible because the universality of the genetic code, meaning whatever is the language that is required to understand for making certain thing is the same for a bacteria, it is the same for us, and it's same for everyone the world over, in the living world over. Okay, so then we are talking about the nitty-gritties, like the entire protein synthesis process kind of has two main parts to it. One is transcription, okay, and then you would have the next part wherein we have translation. Okay, so we also have certain intricate details that we would be talking about, that is mRNA processing. We would be talking about mutations and what is their effect. Some mutations are silent, meaning there is no effect. Some mutations cause missense, meaning you would have something which is being made, but it is not the actual material that you need to be making. Okay, and nonsense kind of stops the entire process of protein synthesis. You can also have extensive problems by you have like an insertion of a few bases which kind of changes the reading frame, and or you miss a chunk of DNA which also changes the reading frame, which can change the code. Okay, so in this case, what we have when we start is, once again, the information in DNA is present in specific sequences of nucleotides. Now what I mean by that is, if this is my DNA strand, I would have, one second, excuse me, I would have A, G, G, T, C, C, A, G, G, C, C, A, T, 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 A, C, C, whatever. Okay, on the opposite side I would have a certain sequence. Now the way in which the sequence of nucleotides are present are, one thing we all know is that two strands are complementary. All right, so the opposite side will have T, C, C, A, G, G, T, C, C, G, G, T, A, T, T, G, G, and so on. Now, if this is the sequence and the way the information is present is in a sequence of every 3 bases, it represents a certain amino acids, which we will see in a few slides from now. Okay, and so they code for a certain protein, and that is the information that they have. Okay, so it's very, very specific. Now DNA is inherited, all right, which means whatever is the information present in the mother cell will go exactly copied, okay, and inherited by the daughter cells. This is the process of mitosis, remember, or meiosis too. Okay, now once you see that you have this sequence and then some technology of transcription and translation sets to work, and then you have finally the protein, you know that this is my final protein and this is what I see. Okay, or it can be an enzyme which is responsible for making of melanin, so skin color, say for example. So that means whatever is present in your genes is finally seen in the physical expression of that gene. So proteins are the links between the genotype and phenotype. Okay. So this process of expression is having 2 stages, like I discussed a few minutes back. One is transcription, the other is translation. Okay. where the information is then made into a protein. Okay, so what Garrett did, he kind of worked on certain disorders, and he worked his way back. If this disorder exists, that means the ability to produce a certain thing is lacking, and that means that ability is lacking because there is a defect in the gene in that person. So he kind of correlated that, and so he thought that the symptoms of an inherited disease reflected the inability to synthesize a certain enzyme. Okay, so we also now need to talk about another set of scientists who came to a conclusion, and how they got to that conclusion we would work our way through briefly. The name of these scientists are Beadle and Tatum, or sometimes it's called Beadle and Tatum. Okay, now they kind of worked on the mold called Neurospora crassa. This is a wild type. It can grow on a minimal medium. Now, what that means? A medium that is made in the lab for bacteria or for fungus, which has minimum things present in it, which means it has some salts, it has glucose as a sugar, okay, no organic matter, okay. So that means there is nothing extra. There is no extra vitamins, there is no extra coenzymes, there is no anything extra present in it. Everything else the organism kind of makes by itself. I mean, if it has a sugar, which is glucose, a pure form of glucose, okay, and some salt which has got nitrates present in it, that means it has got its carbon and nitrogen source. It can make all of its vitamins. You do not have to supply it. Now, on the other hand, a complex medium, what I mean by that is a complex medium would consist not of something that is so specific. It would have, say, beef extract. So that means it has blood, it has protein, it has vitamins, it has heme, it has all of these nutrients that can be accessed by the organism. Or it can have meat extract, or yeast extract, or it can have, say, you would call that molasses. Instead of just sugar, it has got molasses, or it has got some raw form which is not just the sugar, but it has got more additives that are naturally present in that form. Okay, that is a complex medium. Now, many organisms are kind of very, very picky. They need to be given a complex medium because they cannot make these vitamins on their own by themselves. But this particular organism is very very Spartan in its requirement. It kind of can grow in a minimal medium. What they did was they took this particular mold and they bombarded it with X-rays. What is the assumption? Why did they do this? If you bombard it with X-rays, the DNA gets messed up. That means there is a mutation that has taken place. That means there is an inability to produce a certain thing, to produce, say, a vitamin. Okay, now going to the next step on this. So if this is the case, what they would do is they will try to take all of these survivors, okay, and they kind of grow it in a millimeter medium, and they add just one extra material to it. Saying, for example, like I say, if I have a particular vitamin, say vitamin X, I'm talking about, okay, A to B to C to D to this vitamin X. This is how it is made. This is the pathway. And each of these steps need an enzyme. Enzyme 1, enzyme 2, enzyme 3, enzyme 4. Now, this is a known set of steps. Now, what they are assuming is because of this bombardment with X-rays, this has gotten kind of messed up and it can't produce this. If it can't produce this and it is very vital to live, which means in the medium that you are growing it, you have to supply it. Okay, so 1 second, let us see what happens in the next page. Just give me a minute, I'll come back to this. So what they are trying to say in this is if this was a wild type, okay, if it was a wild type, meaning no mutation, it can kind of make this by having a gene for this particular enzyme, gene for this enzyme, gene for this enzyme. Supposing it didn't make it and they supplied it with this ornithine, and so then it made that particular vitamin. Or they supplied it with this, but it still got messed up, meaning this particular gene which makes this particular enzyme got messed up. So if they directly supplied with this, then the organism was able to grow in that medium. Or in this one, they kind of directly had to supply it with the final product because it does not have the ability to convert from here to here. My point was, if it is from A to B to C to D to my vitamin X, like I said, if the mutation because of the X-ray happened here, meaning to this enzyme which got messed up. 1 second, let me just go back. A to B to C to D to vitamin X. Okay, so this one got messed up, and so I would like to be having a medium where I supply this and this organism will grow. Now, supposing this was the enzyme that got messed up, even if I supply this, it will make no use. I have to supply this and it will still make my vitamin X. If this got messed up, meaning the enzyme that produces this enzyme, the gene that produces this enzyme that converts from C to D, if that particular gene got messed up, then I need to supply this. Only then I will get my vitamin X. So this particular experiment, what are the two things that they came to know in this? One, they elucidated a pathway very, very clearly. They knew what changes to what in an entire pathway, and what were the name of all of the components that is being made to give you your final product. And if the particular mutant is not growing, and if you directly supplied it with this and it still grew, you know that this was the gene that got messed up. Likewise for this one, likewise for this step. All of this led them to conclude one enzyme is coded or has information for by one gene that is present in the DNA of that organism. So this gave rise to a hypothesis called one gene, one enzyme hypothesis. Let's just go back and see what I'm trying to say. So that is what he was trying to, was kind of concluded by Beadle and Tatum. One gene, one enzyme hypothesis. Then later on, they modified this hypothesis. Why? For example, let's just take the example of hemoglobin. Hemoglobin. What is the purpose of hemoglobin? Oxygen to be taken and supplied to different parts, right? For this, they need to know what is the hemoglobin protein structure like. It consists of 2 alpha units and 2 beta units. This is my entire hemoglobin. That means I have alpha polypeptide, 2 of them, beta polypeptide, 2 of them. Putting together, I have my final hemoglobin. That means there is one gene that is responsible for alpha polypeptide. There is another gene that is responsible for beta polypeptide. So that means you cannot say one gene is responsible for one enzyme or one protein. You can only say one gene is responsible for one polypeptide. So they modified or tweaked the hypothesis to make sure that it is now known as one gene, one polypeptide hypothesis. This is an important thing to remember. Okay. So we go on to talk about the basic principles of transcription and translation. Okay. So the first thing is RNA is the intermediate guy between the genes and the proteins for which they code. Okay. So which means I have DNA. It is not directly going to be made into a protein. There is a middleman here. It is first going to be made into a readable form of RNA, and then that is going to be read and transformed or translated into a protein. So the first step is transcription. So this is a synthesis of RNA under the direction of DNA. So DNA will first make RNA. The transcription produces messenger RNA or mRNA. Where does this happen? It is different in different living systems. Prokaryotes, it is right there in the cytoplasm because there is no special nucleus. It is only a nucleoid. So there is no special nucleus, only nucleoid region present. Which means in prokaryotes, it is in the nucleoid region, but it is in the cytoplasm directly. In eukaryotes, because eukaryotes have a nucleus which is enclosed inside a nuclear membrane, it is taking place inside the nucleus. Okay. So in prokaryotes, because it is directly in the cytoplasm, as soon as it is transcribed, the mRNA or the messenger RNA, it is immediately translated without any more processing. Meaning, I have this DNA and I am making an RNA, and while the RNA is being made, it is starting to be transcribed. Sorry, translated, I apologize. So I repeat. This is your DNA and I'm making an RNA out of that. Okay, this RNA that is present is then transcribed. I apologize, one more time. This RNA is being translated. Okay. So, the process by which DNA is turned into an RNA is transcription. The process by which the RNA is turned into protein is translation. Okay. Okay, so in eukaryotic cells, the nuclear envelope separates the process and makes it distinct and different. And there is a different timeline. First, I will do this. Second, I will do this. Okay, So in eukaryotic RNA, the transcripts are modified through a process called RNA processing. This is to yield a finished mRNA. Okay. So translation, on the other hand, is the synthesis of a polypeptide which occurs in the direction of mRNA. And ribosomes are the spots where the translation takes place. Remember, the ribosomes are either attached to the endoplasmic reticulum or freely present. And this process of translation takes place in the cytoplasm. This is common for prokaryotes and eukaryotes. Okay, so this is like an outline which just runs home the point again. So this is a bacterial cell, okay, and this is a eukaryotic cell. So you can see that the DNA is just in a nucleoid region and there is no membrane separating it, and so transcription takes place first and an mRNA is made. This transcript is then translated using the ribosomes, which make the polypeptide or the protein. Okay, on the eukaryotic cells, on the other hand, what you have is a nuclear envelope, and this kind of encloses the DNA, okay, which means the transcription takes place here and the mRNA is made. Now this mRNA is a pre-mRNA. It is before it can actually be used. It has to be processed. So when I teach this in my class, I have a kind of a theory that or an example that I use. So what I try to say is I use the example of Voldemort, and I ask my class if they have read Harry Potter, and in that I ask them if they know about the different, the Horcruxes that he had made. So Voldemort had made 7 Horcruxes. What basically it meant was he was making himself invincible, making sure that he is not killed in one go. So it's like saying cat has 9 lives, meaning in order for you to kill him, you have to kill the first one, second one, third one, fourth one, fifth one, sixth one, seventh one. Now what I mean by this is, put it in perspective with respect to our bacteria and eukaryote. Okay, first, bacteria are very small in size, okay, and their DNA is constantly exposed to UV, and they have most of the time exposure, and they also keep getting mutations. They keep changing. So this is like kind of like a fact. And then they also have very good amount of DNA repair enzymes. On the other hand, okay, you have eukaryotes which have DNA inside the nuclear membrane, and you have them as chromosomes. Okay, so the number of DNA is like a lot more. Okay, so these chromosomes have a lot of DNA in them. Okay, so suddenly there is like a big leap in the quantity of DNA from a prokaryote to a eukaryote. Granted that they are bigger organisms, they have more complex functioning systems and so on, and yet there is lots of DNA that is kind of not really having or making sense or having any think that you can make some useful material from that. Meaning, lots of junk DNA coding for nothing, meaning nothing. Now, on the same note, when mRNA is being made from it, okay, so my point is, supposing mRNA is being made from this, what is being made actually is, supposing this is my DNA, okay, and only certain parts of my DNA are kind of useful, and the mRNA that is made out of it, I'm making this long huge thing, I really have this with me and I have no use for it. So what the cell does is kind of removes those guys and then reattaches these guys in a way that it sort of and makes it into a edited version. That's the right word for it. Okay. Now I can give another example, like during a wedding. Okay. Now during the wedding, I mean, it's like for an entire day, and you would have the videographer taking video of just about everything that is happening on that day. And yet, when he gives you the video, it probably is just about an hour or two long. Okay, so he's kind of edited a 12-hour long session into a 2-hour long video that you can see. So he's basically spliced the video, cut certain sections, removed it, and taken only certain sections and put them together. Okay, so this particular process, okay, is one part of mRNA processing. So the DNA that is there, it first makes an mRNA. It is not called mRNA that is ready to use. It is called pre-mRNA. It has to be processed, and then you have editing taking place. It has a certain set of things to be done, which we will talk about in the next slide. Okay, just a second. Let me just go back and show you what is in that next slide that I would like for you to know, and then okay, so the details of the mRNA processing will be discussed when I reach that slide. Let's just finish a little bit more description about the process of transcription and genetic code and the central dogma. So, the central dogma is like, what is the chain of command? Who is the topmost person of authority? Okay, so the molecular chain of command is like the person who has the information, and he kind of signs off on that whatever information he has, and he passes the information to his secretary, who say types it up, and this secretary then sends it over to the supervisor, who then kind of gives it to the actual person who works on it and makes the final product. So I've just given you an analogy as to how this would work. So the person with the information is the DNA. The secretary is the one who is kind of giving that information, and the supervisor is the one who makes sense of it and gives it to the person who works it out and gives you the final product. All right, so the RNA would be the supervisor, and then the ribosomes that are involved in it would be making the final product. Okay, so then we are going to talk about the genetic code. Before that, I just want to reiterate another point. The molecular chain of command for all life forms is always from DNA to RNA to protein. There is only one place where it is different, meaning there is a specific RNA virus that first has to make a DNA, and this DNA then makes the mRNA and then the product. And this particular, the roles are like kind of reversed. RNA is the main guy in this specific case only. Okay, so we move to the part of genetic code and how it is come to be. Okay, so the first thing, we have 20 amino acids. Okay, we have 4 bases. What I mean by that is 4 nitrogenous bases: A, T, G, and C. Okay, now because it is RNA, I would like to rephrase that as A, U, G, and C, because thymine is never present in RNA. Instead, you have uracil. Okay, if one base represented one amino acid, we would have code for just 4 amino acids. What about the rest of the 16? Yeah, so that is not ideal. So if I say we have 2 bases in combination representing 1 amino acid, meaning I have A, U, G, C. Let me call that 1, 2, 3, 4. So I can have a combination of 1 to 2, 1 to 3, 1 to 4, 2 to 3, 2 to 4, and 3. Excuse me. So I can actually have a code of 4 raised to 2 combinations, wherein 16 combinations with 2 bases at a time. I mean, you can write this, all of the different combinations that you can have with 1, 2, 3, 4 or AUGC. You can have 4 raised to 2 set of combinations, and that would be having code for 16 different types. That means I have code to represent only 16 amino acids. What about the other 4? So that is not suitable. Instead, if I have 3 bases at a time representing a code, okay, 3 bases or representing a code, which will mean I can have 4 raised to 3 combinations, which is 64 combinations possible. And in these 64 combinations, I can definitely accommodate my 20 amino acids. It seems excessive, but still I have the ability to accommodate the expression of all the 20 amino acids. Okay, and so genetic code is a triplet codon. Okay, a triplet codon means you will have groups of 3 representing 1 amino acid. Now, for example, if I have A, U, G, G, A, C, C, A, U, G, okay, this is one amino acid, this is another amino acid, this is another amino acid, see, this is another amino acid. So it's very important to remember the math behind this. Okay, my point is, you can have one amino acid represented by 3 bases. So if I have 30 base pair long strand, I can represent 10 amino acids, or my polypeptide can be 10 amino acids long. Does that make sense? Of RNA. Alright. So if I have 100 and so see a polypeptide with 100 amino acids, my RNA has to be 300 base pair long. Does that make sense? So one amino acid is represented with a sequence of 3 bases. This is something that you must remember, so you can kind of convert. So if they ask you how many amino acids do you need, or how many base pairs do you need, vice versa, whatever the problem can be, you can answer. Okay, you're kind of equipped to answer that. This is one of the things. Second is, when we have a strand, just let me to explain this sentences, this template strand and the others, just to explain that, what happens is, this is my DNA. It is 2 strands. Sometimes this is, let me just call this strand 1 and this is strand 2 of DNA. Sometimes strand 1 can be the template to form RNA, and so it will be called the coding strand. Sometimes strand 2 can be the template and it will be called the coding strand. So if one is the coding strand, the other one is the non-coding strand. For some polypeptides, it will be strand 1. For other polypeptides, it will be strand 2. So for whatever polypeptides, if the strand 1 has been the template, it will always be that strand. And for others, it will always be the other strand. Okay. So during translation, the mRNA based triplets called codons are read in the 5' to 3' direction. By now, my class, hopefully you know what I mean when I'm saying 5' to 3'. Okay. So for that, just a refresher one more time. If you have the sugar, the pentose sugar, the position 1 is taken up, the hydroxyl group bonds with a base. Okay. The position 5 bonds with a phosphate group. Okay, this is the position 5. So this is position 1. So the 3' hydroxyl is the only one that is free for future extension or elongation of strand, which means the extension of a strand is always from the 5' to 3' direction. And so in an mRNA also, when you're reading the codons, you have to read it from the 5' to 3' direction. Each codon specifies an amino acid to be placed in that specific spot along the polypeptide. Okay, so in this case, what he is talking about, okay, he has this particular DNA, okay, this section of it represents gene 1, this section of it represents gene 2, this section of it represents gene 3. So, excuse me, if you have this particular strand we are talking about, okay, DNA, it has coded for this strand, which means this has been the template, or this is the coding strand and this is the non-coding strand. And if you notice, it is exactly complementary to the bases that are present in the template strand. If there is an A, it has to be a U, and instead of a T in an RNA. If it is a C, it is a G. C is G. A, U, A, U, A, U, G, and so on. Okay, so you will have a complementary strand of mRNA being formed for that one section of DNA. It does not mean the entire length of the DNA is going to be transcribed into an mRNA. Just that particular section, that chunk of DNA, that strand, one strand will serve as a template. So I have my mRNA. Please note it is from 5' to 3'. So the first three stand for a specific amino acid. The second three stand for another amino acid. Third, another amino acid, and so on. Okay. Now, like I said some slides back, genetic code is a sequence of 3 codons in the 5 read in the 5' to 3' direction on the mRNA. Each of those codons represent one amino acid. So there are two more facts to remember about the genetic code. First, we already know it is codon is groups of or sequences of 3 bases at a time. It is read in the 5' to 3' direction. Okay, one codon represents one amino acid. Okay, so genetic code is redundant, but it is not ambiguous. What he means by that? Okay, so he says, if you notice, we have 20 amino acids, which means I only need 20 codons. But in this chart, you will notice, if they are talking about leucine, I have this, this, and this representing leucine, which means there are 6 names for leucine, or 6 codons for leucine, the amino acid leucine. So the genetic code is redundant. Kind of like I sometimes take the example of a certain name of a student in my class. Let's just call John Smith. So Mr. John means that same person. Mr. Smith means that same person. So if it is John Smith Wilkerson or something, so Mr. John represents that person, Mr. Smith represents that person, Mr. Wilkinson represents that same person. So that one person can be coded for or called by 3 different names. Genetic code is redundant, but it is not ambiguous, meaning if John Smith Wilkerson is the name of this one person, it will still be the name of that person only. It will not be the name of someone else. Excuse me. So my point is, if these are the codons for phenylalanine, okay, they will always be representing phenylalanine. You cannot be for something else or something else. It is not ambiguous. That's what he's trying to tell you. And in this chart, if you notice, there is one which is shaded in green, AUG. Remember, this is your 5', this is your 3' end. So the way you would read it, excuse me, let me just make this blank and then we'll start. Okay, so okay, so my point is U, and that is this one. So if this is what is present, or supposing I have C, G, A. So you search for C in this. C, and then the G, so CG, and then the A, so CGA. That's it. So that is how you search for the codon, and that is how you identify that particular codon representing that particular amino acid. This is how you read this chart. Okay. In this chart, if you notice, AUG, which means the A, and then the U, and then the G, representing this particular amino acid called methionine, is always used to start a translating a particular amino acid, sorry, a particular polypeptide. So this is always going to be the start codon. Okay. And if you notice, UAA, UGA and UAG are codons which do not code for any amino acid. Okay, so out of the 64 out of the 64 codons that are present, 61 of them represent the different amino acids. These three represent stop, and you'll get to understand what it means. It kind of is in the end of making a polypeptide, wherein it kind of indicates to the protein synthesizing machinery to stop adding any more amino acids to that particular polypeptide chain. Okay, so another important part to be noted about a genetic code is universal. So the same code is shared by the simplest bacteria to the most complex animals. So because of this particular feature, you can actually take this particular gene and put it into another species and it will still be read and you will get a product. This is the basis of genetic engineering. Okay. For example, they have taken the gene, firefly gene, okay, firefly gene, which has the glowing abilities, and insert it into a tobacco plant, which is now having the ability to show bioluminescence. Another one wherein you can see jellyfish gene, which shows bioluminescence, is inserted into the genes of a pig, which shows the ability to be bioluminescent. Okay, now we work on the details of transcription. Okay, so the molecular components of transcription. What are the things that you need for the whole process of transcription? An enzyme. This enzyme is called RNA polymerase. See, anything that can polymerize and make a polymer of RNA is RNA polymerase. That's how you roughly calculate the meaning of it and remember it. And at all times, instead of thymine, wherever there is a need to place a T, a uracil will be placed instead. And the whole process starts by a promoter sequence that is recognized by the RNA polymerase, which means on the DNA, this RNA polymerase kinds of sits and zips through, and where it can see a sequence of DNA called TA, that is where it gets an indication to say, oh, I have to start transcribing this region. Then you have something known as terminator or stop codons, and then the entire unit is called the transcription unit. Okay, I would like for the class to first watch a video and I can continue to talk beyond this step.
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured. The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA. As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus. The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the large subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases, called an anticodon, that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids. For proteins that will be secreted from the cell, like insulin, the ribosome docks on the rough ER, and the protein grows into the ER compartment. The new protein molecules are packaged in a vesicle that is transported to the Golgi apparatus, where many proteins are processed. However, insulin is packaged in a vesicle that leaves the Golgi and is then processed. Proteins secreted from the cell are shipped to the plasma membrane. Here, insulin is secreted from the pancreas and begins to regulate the student's rising blood sugar levels.
Okay, so giving that video as a start will give you a kind of perspective about what I'm talking. Okay, so the transcription unit is ready to go through the first step in the protein synthesis of transcription. Okay, so the first thing what it does is this would be your DNA double-stranded, and the promoter region, which is the region which is recognized by the RNA polymerase because it has the sequence of TA written, called the Tata box. And this is your RNA polymerase enzyme which kind of recognizes that part and says, okay, this is where I have to start to transcribe or make a mold of or template of one of the strands. Okay, so it starts to make a RNA transcript. Okay, and once the RNA transcript elongates, I have an RNA that is being made. Now, if this is in prokaryotes, as soon as it's made, it is going to be translated. But if it is in eukaryotes, it has to be if it is in eukaryotes, it has to be processed. Okay, so this RNA transcript that is being made, it falls off after a certain spot where it kind of terminates the process of making the RNA from the DNA of, or from the gene in the DNA. Okay, so the synthesis is divided into 3 stages: initiation, elongation, and termination. So it gets initiated when the RNA polymerase recognizes the TATA boxes and kind of starts the whole process, and then elongation, and finally it can reach sequences which kind of signal the termination of the RNA transcript. The RNA polymerase falls off, leaving the RNA transcript that is being made out for the next step of processing. So during the process or the time of processing, the RNA is essentially having a GTP added to it. Let me just go back, I'm sorry. Okay, it has a GTP or a guanosine triphosphate added to it in the beginning, and that is called the cap. And then in the end, it has got a lot of A's being added. A. That is called the poly A tail. And then there are certain regions which are not translated, and it is called the untranslated region. Okay, and then you have the start codon, or the codon which represents the AUG, and then the stop codon can be UGA, UAG, and UAA. These can be your 3 stop codons. Okay, in between it has a lot of spots which are not really required. Remember, a few slides back I was talking to you about lots of junk DNA which have to be spliced. I also talked to you about the wedding, wherein the videographer kind of slices out the unimportant parts and uses only the interesting part to make the final wedding video. Kind of like that. They are regions in the DNA. Okay, these are called introns and exons. Okay, now introns are interfering sequences. That's what I like to call them, so that you understand the difference between the two. Exons, I would like to call them as eventually expressed. Those genes are eventually expressed. So what is the purpose of adding the cap and the tail? This helps in the export of the mRNA, and it protects it from the hydrolytic enzymes, and it also helps the ribosome attached to the 5' end to kind of get itself fixed to get the whole process started, the second step of translation started. Okay, so once again, untranslated region is UTR. Intron is not coding sequences. I call it interfering sequences. And exons, I call it eventually expressed regions. And then you had the DNA kind of being spliced out, and the small molecules or spliceosomes that were involved in the whole process are sRNA or sRNP. They are called as NERPs in short. Okay, or ribozymes. If you remember, when I was doing the chapter on enzymes, I had told you 99% of all enzymes are proteins. The 1% that is different is the spliceozymes, which are ribozymes. They are made of ribosome. They are made of RNA. So small nuclear RNA. So these function as the enzymes. The ability to hydrogen bond and specifically, and also encourage catalytic activity. Because it is single stranded, base pairing is possible, and some of the bases have functional groups that help in the catalysis. So then the cell interprets this genetic message and builds a polypeptide. The message is a series of codons on the mRNA. The interpreter is the tRNA or the transfer RNA. Okay, the function is to transfer amino acids from the cytoplasmic pool of amino acids to a ribosome. The cell always keeps a stock of amino acids in the cytoplasm for this purpose. Okay, so the structure and function of transfer RNA. It kind of sort of looks like a clover leaf, and each of them carry a specific amino acid on one end, and they have an anticodon on one of the ends of the clover leaf. Okay, that can base pair with the complementary codon of the mRNA. What I mean by that is, this is my mRNA, my tRNA, this three bases on the tRNA kind of are complementary to the bases that are present on the mRNA or the codon, and that kind of helps them identify which amino acid has to be brought for the next step in bonding to the existing chain of polypeptide. Okay, so if you notice here, this is the clover leaf structure. You would have the 5' to 3', so it's kind of read in this fashion. So this is my 3', and I have my amino acid here. This is my 5', and this, and this is exactly complementary to my 5' to 3'. This, and this codon. Okay. When they bond, they kind of are capable of letting go of this amino acid, and there is a catalysis of that into the increasing chain of the polypeptide. Accurate translation requires two steps. Okay, first it needs a tRNA and it needs an amino acid, and the enzyme is amino acyl tRNA synthetase. And the second thing that it needs is the correct match between the tRNA anticodon and the mRNA codon. Okay, ribosomes facilitate the coupling of the tRNA. Let me just go back once again. Ribosomes facilitate the specific coupling of the tRNA anticodons with mRNA codons in protein synthesis by providing a platform. The two ribosomal subunits are made up of proteins and ribosomal RNA. Okay, so my point is, first the mRNA is kind of binding here. Okay, the first amino acid will come and sit here. Then the mRNA kind of pulls it, so it has gone here, and then the newer amino acid will come here. At this point, this will go on to get up and escape. There will be a third amino acid carrying tRNA that will come here. So it's like a sequence in which you will have them being added. So there are these three sets of 3 sequences. It is called, the middle one is called the peptidyl binding site. The extreme left one is called the exit site. The extreme right one is the amino acyl tRNA binding site, which brings in fresh amino acid. Okay, so every time you would have a fresh tRNA coming in, the old one kind of escapes, and this entire polypeptide kind of attaches to the new one, and this one escapes then after that. Okay, so you would have the way it is organized is, let me just, the way it is organized is, it kind of is the start codon. Let me just put this. Okay, so this is where it would bind, dock itself, the mRNA. This is the start codon bringing the AUG. So I have the AUG containing bases here. Okay, and then the CAU bases here, and then that brings in the amino acid. It's kind of like this, and this will initiate the whole process. Then I have the bigger unit of the ribosome coming and fixing to the smaller unit, base unit, and it kind of, the mRNA moves kind of like being a carpet being pulled from under you. So you would have the next three being ready to be read. I would have the next amino acid coming here. At this particular point, this guy will transfer it to 2, meaning I want to say 1 first comes in. Then when 2 comes in, 1 gives it away to this one and 1 is out. So first in, first out. That's the kind of system. Then when 3 comes in, 2 gives the responsibility of holding all the polypeptides, including the amino acid that it contains, and this exits out. Then when 4 comes, 3 will give it to 4. When 5 comes, 4 will give it to 5, and so on. Okay, so the ribosome has 3 binding sites for the tRNA. The P sites holds the tRNA that carries the growing polypeptide chain. The A site brings in the next amino acid, the new ones. The E site is the exit site where they are discharged and leave the ribosome. The attachment is not like the amino acids to a polypeptide chain, but it's like a bead at a time. It is not like the amino acids. Oh, I'm sorry. Let's just go back again. Okay, the attachment is not like amino acids can be put into a polypeptide chain. Like I have this chain and I keep adding to that. No, it is not like that. What happens is, you think of a skewer stick. So if you poke onto the cheese, the first cheese finally finds its way to the end, and the newest cheese at the pokey end. So then you'll have this again, 3rd, 4th, 5th, 6th, 7th, kind of like that. Okay. So the 3 stages of translation is initiation, elongation, and termination. All of these require protein factors that will aid in the translation process. Okay. Okay. This video hopefully will be helpful.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay, so the step where you have the elongation of the polypeptide chain, during this time the amino acids are added one by one to the preceding amino acid. Okay, each addition involves proteins called elongation factors that occurs in 3 steps. First, they recognize the codon. Second, there is formation of a peptide bond. Third, there is translocation of the peptide bond and the tRNA moves off. So the first step, wherein the fresh amino acid is being brought to a mRNA that is getting translated. Okay, then you have the second step, wherein the existing polypeptide is added and you have it forming a peptide bond with the newer amino acid that just came in. And at this particular point, this older tRNA that is present is getting ready to exit. Okay, at this point it is ready to have the next amino acid containing tRNA to come and sit at the A site. So this is the cycle with which you would have translation taking place. The termination of translation occurs when you have like a stop codon in the mRNA, and that reaches the A site of the ribosome. At this particular part, what happens is there is no amino acid that the tRNA can bring in. So the A site accepts a protein called a release factor, and instead of it adds a water molecule instead of an amino acid. This releases the polypeptide and translation assembly comes apart. Okay, now what is a polyribosome? This is like a term that you have to know. A number of ribosomes can translate a single mRNA simultaneously. What I mean by that is it can be like, for example, I have, say, a paper and I ask the entire class to make copy of it. What happens is one person has started it and then he has reached, say, page 3, but there's another person who started a little after him, he has reached page 2. Another person who started after him, he has reached page 1. When this person goes further, it will kind of make way for another ribosome to start making, meaning one mRNA can be used by an entire set of this entire set of this ribosome translation unit to keep making a lot of polypeptide, which at any point in time resemble a polyribosome, wherein you would have the polypeptide copies being made, and all of them are in different stages of being made. Okay, and that looks like trucks in a convoy. So you would kind of see this in this particular place. What is happening is this particular ribosome is done with making the polypeptide, so it's done, but it has reached the end. There is something that is just about starting, and it's going to start making the polypeptide. So a mRNA strand having multiple ribosomal units in the process of making polypeptides, that resembles something known as a polyribosome. And this is what you can see under an electron micrograph. Okay. Often translation is not enough to make a functional protein. After that, you have to have like modification after the translation. Sometimes the completed proteins are targeted to a certain specific site in the cell. Okay, so that part is done. Now we are going to deal with the part of mutation, and we're going to talk about how it affects the expression or the process of translation, how it affects. Okay, and is it always so bad? Okay, so mutation, first remember, is like the most important thing, and it is like the ultimate source of new genes. It is responsible for diversity. Mutation is not all bad. Okay, and you can have mutations in different forms. One, you can have physical changes to the DNA structure, and that's a mutation, or chemical changes to DNA structure. But you can have different kinds of mutations, which is point mutation, which means suddenly you have one extra base added, kind of squeezes itself in between, or one base less. Okay, or it is substituted for a base. Instead of say an A, there is a T instead. Okay, if this happens in a gamete, then the future generation will have that wrong information in them, in their cells. So a series of such wrong informations, if they are not making a specific destructive change, they can continue to be accumulated, and over a period of time you will have the organisms evolving. Now I would like to talk about what I mean when I say point mutation in case of one base being deleted, one base being added, or one base being substituted. So point mutation, what are the different kinds of point mutation? One base deleted, 2, one base added, 3, one base substituted. So the old fox can hop and get the supposing. Now what I'm doing is, instead of just randomly putting the A's and the D's and the G's and the C's, I'm using it like a sentence to make it a little more easy for my class to understand. Okay. Each of this represents an amino acids. Like these are 3 bases representing an amino acid. Okay. Imagine now that you end up having one extra base being added. Say for example here A. So now, with respect to the entire thing being read, you would have the as the old, but afo exca naho pan de ebo. So you see, it kind of changes the reading pattern. This is one option. Okay, let's try the other option. Supposing one of them gets deleted. Okay, now let's see what is happening. The olf, ox, and opa, and it, help, something else. So it changes the reading frame again. So in point mutation, if there is a deletion or an addition, it kind of changes the reading frame. Okay, now another option. If I have, instead of a deletion or an addition, a base is being substituted. So I'm going to put it like, okay, now that was a D, but I put a B, right? Meaning I changed the base, or there was a base substitution, right? Which means the olb, fox, can, hop, and if you notice, it really did not change it much. Now what is more important in this is what has happened to this. If this represents the same amino acid, how, remember, genetic code is redundant. If it is redundant, meaning the amino acid, say glycine, was old and olb, that means I will still have glycine being put here, and I will still have the same polypeptide. So this mutational change that took place did not really make any change to my protein, and this is silent mutation. On the other hand, okay, from old, it has become olb. Okay, if this can mean another amino acid and it changes the structure, it is called a missense mutation. Okay, third option. Okay, instead of old it became olb, and this was a stop codon, and the protein synthesis just stopped right here. These things were not even read. That is a nonsense mutation. Okay, let's see what happens in the next few pages. So this is where I was talking to you about, supposing I had a mutation wherein I had a substitution of C instead of U, but it still represents my phenylalanine. That is silent mutation. instead of UCA, I got a C here. Sorry, UUA, I got a UCA. So I got, instead of leucine, I got serine. That is missense mutation. Okay, instead of UAC, I ended up having a mutation which said UAA. That is a nonsense mutation because it stopped it. So that's nonsense mutation. Okay. Okay, so here he is giving the examples with certain bases and he is comparing it to sickle cell anemia. Okay, in your wild type hemoglobin DNA, this is what is there. And now I have a mutation wherein, okay, instead of T, I have an A. And so what happens is I end up having a GAA in my normal hemoglobin. Instead I have a GUA, and this gives me valine in that. So this is like a missense mutation, remember. Okay, but it completely changes the structure of my hemoglobin and I suffer from this. So this particular mutation has happened in just one base in the 6th amino acid, by the way, of a polypeptide, which makes hemoglobin. Okay, so production of an abnormal protein, you can have base substitution wherein it replaces one nucleotide and its partner with another pair. It is silent if there is no effect. If it is missense mutation, it has little effect maybe, or maybe if it is a similar amino acid, or it is in an unimportant region. Now what I mean by this, remember we have, say for example, the fox can hop off the box. Okay, supposing I had a mutation here and instead of this I ended up getting born. Okay, but it is like very much downstream. Most of my protein is anyway done. It was almost in the end that it messed with it. It might have very little effect. That's one option, because it was downstream and in an unimportant region. Maybe. Or if a similar amino acid was added. What I mean by that, if there was supposed to be an acidic amino acid in that place, and I still have an acidic amino acid but a different one, I'm still okay. Or if it was supposed to be a hydrophobic amino acid, I kind of lost that amino acid but I have a similar hydrophobic amino acid, I might still retain the properties and structure of the polypeptide that I'm dealing with. So it might have a little effect, it might be having extensive effect deleteriously. Okay, the third option is nonsense mutation, when the translation process completely stops abruptly and I have a non-functional, half-formed, partially formed protein. Then again, this is my silent, wherein if you notice the GGU and GGC represents the glycine, so it's not making any difference. This is missense, wherein if you notice GGC was glycine but instead I got AGC and it is serine. This is missense. And the third one, if you notice, instead of GGC, or for example stopping at this particular part, it kind of sort of stopped. Instead of AAG it did UAG and it kind of stopped it right there, and I didn't have any functional protein. Okay, so that is one part of it. But remember, all of that I was dealing was maybe just one base more, less, or changed. In this case, it's an insertion or deletion. An entire segment of DNA is either added or lost. This alters the genetic message because it alters the reading frame, and it's called frame shift mutation, wherein you will have extensive missense mutation, total gibberish being read. Therefore, nonsense protein causes premature termination. Improperly grouped will cause extensive missense mutation. Okay, so that's what he was trying to talk about. It kind of stopped right in the beginning when there was an insertion of this particular base. Now let me just go back. Okay, insertion of this particular base here, you ended up having, because this kind of shifted and caused immediate nonsense. On the other hand, I can have one extra wherein I start to have gibberish amino acids which don't make any sense to the function of my protein that I wanted. And you can instead have no frame shift but just one amino acid is missing. Sometimes if it is very much downstream, you can have 3 base pairs being deleted. That's a possibility, and it can change the outcome. It may not change the outcome depending on how important that particular amino acid was to the structure of that protein and the functioning of that protein. So mutagens, on the other hand, that we are talking about are the ones that can generate a mutation. Okay, so mutagens are something that can generate a mutation. They can be spontaneous or induced. Spontaneous means naturally they can occur, or induced means I can actually bombard it with X-rays or something, like we did in the Neurospora crassa experiment. Okay, so mutagens could be physical mutagens, meaning physical agents that are present are high electromagnetic radiation, UV rays, X-rays, gamma rays. Okay, or chemical mutagens actually have bases which look like the original bases, so they kind of are like doppelgangers. Or they resemble the bases. Okay, and therefore there is improper base pairing. It alters the helical structure. Most mutations most mutagens are carcinogenic. They are capable of inducing the kickstarting the process of cancer. In bacteria and viruses, I apologize. In the previous slide, we kind of finished talking about the protein synthesis. We finished talking about the different mutations that were possible, and also the mutagens. So this is like just summing up the whole process. Okay, so in bacteria, both processes of transcription and translation is simultaneous, whereas in eukaryote both processes are distinct and separate. In archaea there is similarities to prokaryotes and eukaryotes, but it's a totally different ball game, and we are not discussing that anymore, except for the protein synthesis in bacteria and in eukaryotes that was discussed in this chapter. Okay, we finished with this chapter, and I hope this was helpful.